Good morning. It's August 13th, and this is your daily brief in science. Here's everything you need to know. A recent study published in The Lancet Gastroenterology and Hepatology raises concerns about the impact of artificial intelligence in colonoscopy procedures. The research analyzed 1,442 colonoscopies performed in Poland, comparing adenoma detection rates before and after the introduction of AI-assisted polyp detection systems in 2021. The findings showed that the adenoma detection rate dropped from 28.4% prior to AI use to 22.4% afterward. Meanwhile, procedures using AI assistance achieved a detection rate of 25.3%. This decline suggests a potential de-skilling effect, where reliance on AI tools may reduce endoscopists' ability to identify precancerous growths independently. Experts suggest that cognitive offloading, or depending on AI for critical decisions, could hinder visual recognition skills and decrease vigilance. Continuous use of AI decision support systems might also diminish motivation and focus among endoscopists during procedures. Omer Ahmad from University College London emphasized the need for performance monitoring guidelines and educational programs to address these risks. The study advocates for new training frameworks that balance AI utilization with maintaining essential procedural skills. Dr. Markin Romancic, a co-author, noted this is the first research indicating a negative impact of AI on healthcare professionals' performance in a patient-relevant task. However, the study's focus on experienced endoscopists and its observational design limit broader applicability, highlighting the need for further research involving less experienced practitioners. The AI tool in question was designed to identify precancerous polyps, aiming to improve early cancer detection and removal. Overall, the study underscores the importance of understanding how healthcare professionals and AI systems interact to optimize patient outcomes. A new treatment for a chronic lung condition has just received FDA approval, marking a significant milestone for patients suffering from non-cystic fibrosis bronchiectasis. This condition affects approximately 500,000 people in the U.S. and has lacked targeted therapies until now. The drug, brensocatib, marketed as Brinsupri, is the first approved treatment that specifically targets neutrophilic inflammation, a key factor in disease progression. It is also the first dipeptidyl peptidase 1, or DPP1, inhibitor authorized for a neutrophil-mediated disease. In Phase 3 clinical trials, Brinsupri reduced annual exacerbation rates by up to 21% and slowed lung function decline, especially at the 25 mg dose. The ASPEN trial, the largest for bronchiectasis, showed significantly lower exacerbation rates in patients receiving the drug compared to placebo. Common side effects noted during trials included upper respiratory infections, headaches, and rashes, indicating the importance of careful patient monitoring. The manufacturer, Insmed Incorporated, is positioned to expand access through its existing infrastructure, with plans to seek approval in Europe, the UK, and Japan by 2026. International regulatory submissions are underway, reflecting the higher prevalence of bronchiectasis in those regions. Experts like Dr. Dorina Drizzo-Harris see this approval as a potential paradigm shift, addressing a root cause of exacerbations and setting a new standard. The approval also opens new investment opportunities in respiratory therapeutics, as Brinsupri enters a previously underserved market. Researchers at Arizona State University have developed NASRED, a portable diagnostic device capable of detecting infections like COVID-19, Ebola, AIDS, and Lyme disease from just a single drop of blood. This device costs approximately $2 per test and provides results within 15 minutes, making it suitable for emergency and routine healthcare settings. NASRED's sensitivity is about 3,000 times greater than the ELISA method, enabling it to identify disease-related proteins at concentrations 100,000 times lower than standard laboratory tests. The device is designed to be user-friendly and portable, requiring no specialized training for operation. Its affordability and ease of use make it particularly valuable in low- and middle-income countries where access to reliable diagnostics is limited. The technology could improve early detection and intervention, especially among hard-to-reach populations such as injection drug users. Future plans include miniaturizing and automating NASRED, which could allow for use in home settings and expand its application to chronic illnesses and cancer detection. NASRED aims to reduce diagnostic errors that contribute to nearly 800,000 deaths or disabilities annually in the U.S. by facilitating early diagnosis. A study published in ACS Nano highlights its ability to distinguish COVID-19 from other diseases, aiding clinical decision-making and infection control. Overall, NASRED represents a significant advancement in medical diagnostics by combining speed, sensitivity, and affordability. Its simplicity allows healthcare workers with minimal training to operate it, which could greatly improve public health outcomes in resource-limited areas. Preliminary tests indicate that NASRED can achieve sensitivity comparable to established molecular tests like Abbott 1D Now for various diseases. A recent study from the University of Hawaii at Manoa has found that maternal obesity prior to pregnancy can lead to autism-related behaviors in offspring. The research attributes these effects to lasting epigenetic changes in egg cells that occur before conception. This emphasizes the importance of maternal health before pregnancy and suggests that early interventions could reduce risks associated with obesity and autism spectrum disorder globally. Using an in vitro fertilization model, researchers isolated the effects of maternal obesity prior to conception from those occurring during pregnancy. The study has been accepted for publication in the journal Cells, highlighting its significance for understanding early influences on brain development. Led by Professors Alika K. Monakea and Monica Ward, the research shows that these epigenetic changes can profoundly affect brain development independently of maternal contact after conception. Behavioral assessments in adolescent male mice revealed impaired social interactions and repetitive behaviors, traits linked to autism. These mice also showed increased expression of the short Homer 1A isoform, associated with synaptic dysfunction. The male offspring exhibited behaviors such as impaired social interactions and repetitive grooming, characteristic of autism spectrum disorder. While the study identifies an association, it does not establish a direct causal relationship, and further research is planned to explore potential interventions. Recent archaeological discoveries in England reveal that 2 individuals buried in 7th century cemeteries had ancestors from West Africa, challenging previous notions of early medieval England's cultural isolation. DNA analysis from remains in Updown, Kent, and Worth Matravers, Dorset, shows genetic ties to present-day West African groups such as the Yoruba and Menda. Duncan Sayer from the University of Lancashire noted that Kent was a key point of contact with continental Europe during the 6th century, suggesting these findings are part of a broader network of influence. These discoveries indicate that migration and cultural interaction were more common than previously thought during this period. The evidence supports the idea that trade routes connected Britain to distant regions, including West Africa, through complex networks involving merchants and travelers. It is possible that individuals from West Africa reached Britain via trade routes linked to the Byzantine Empire's reconquest of North Africa in the 6th century. Both individuals were buried within their communities, with no signs of slavery, implying they were integrated members of society. Experts argued that these findings challenged the perception of the 7th century as a culturally isolated dark age, highlighting its dynamic and interconnected nature. The research, published in the journal Antiquity, advances understanding of early English population diversity through genetic analysis. The mitochondrial DNA was Northern European, suggesting the West African ancestry likely came from a paternal grandfather. Researchers cautioned that only 2 of 274 genomes analyzed showed sub-Saharan ancestry, so broader conclusions should be made carefully. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.